Natasha, are you here? No. Anyway, we'll still begin. Okay, so let's see. Where were we? We are talking about vision. And actually, let's go back, if we can, to the outline in a minute. Making a mistake. We're talking about vision, and we are getting towards the end of talking about the responsive properties of neurons at various levels of the visual pathway. We talked about the anatomy a little bit, about the pathway itself. And we then talked about what goes on at the level of the ganglion cells. Then we, that's basically, I guess, where we ended. We ended right there, talking about this feedback pathway from the, oh wait, no, we didn't stop there. We got ourselves all the way up to the cortex. And we talked about the fact that there's this feedback pathway from the cortex back to the thalamus that can act to cause positive feedback and to reinforce a weak or noisy or low contrast visual pattern. As I said before, there are these backward pathways almost everywhere in the visual system. So you have a big forward pathway projecting information from the outside in. You also have a reverse pathway projecting information from the inside out, from higher levels to lower levels. The exception to that, is the level of the optic tract itself. One does not have stuff going from the lateral geniculate back to the eye, or at least not to any appreciable extent. But the rest of the pathway, from then on, you do have that happening in a substantial way. Okay, the last level that we want to talk about, we're really tracing our way through the so-called ventral stream now. We just talked about V1. And I'm going to skip for the moment, skip V2, V4, and so on. We'll come back to V4 later on a little bit. And I want to jump all the way to the end of the ventral stream. Remember, the ventral stream is a stream that has to do, it's the so-called what pathway, and the dorsal stream is the so-called where pathway, and we're talking right now about the what pathway. And so what do you see if you record in IT, or intratemporal cortex? It's referred to both ways, IT and intratemporal cortex. Well, the picture is very, very different from what it was in V1. In V1, cells like little oriented line segments at specific locations in the visual field, or specific locations on the retina, projected onto specific locations on the retina by the lens of the eye. In inferotemporal cortex, the picture is entirely different. In inferotemporal cortex, you find cells that respond to meaningful stimuli. When they were first discovered, these experiments were done on monkeys. You would find cells that responded to a monkey's paw or face of another monkey, or some specific investigator in the lab, a human investigator in the lab, something specific that the monkey being recorded from knew. And these cells were quite selective for those things that they would respond to. They were probably not perfectly selective, but they were quite selective. These cells have been studied a great deal, and this was from a much later study from the original ones. This was a cell that responded, in this case, responded to faces, but responded not just to faces in general, but to these particular faces. There were two different cells that were being investigated here. One of them responded to this guy. Sorry, that's not true. The cell responded preferentially to this guy and not to that guy. One cell responded to this guy and not that guy. It happens this guy is the head of the lab, and that's one of his graduate students or something like that. And so this shows the amount of firing, the rate of firing of the cell to these various images. So if this guy looks at the monkey, or the monkey's looking rather at this guy, you get that amount of firing. If the monkey's looking at this guy, you get that amount of firing. Now, these guys don't look terribly different from one another, but they look very different. This cell thinks they look different. Moreover, at least according to this paper, the selectivity remains even if you change the orientation of the individual. So here he is upside down, still selectivity. Here he is not smiling. Here he is smiling. Here he is dressed very differently. Doesn't make any difference. The monkey is recognizing, that cell in that monkey is recognizing and is firing selectively to this particular guy, no matter what his particular circumstances of presentation. Now, in actual fact, I have some reason to doubt taking this totally seriously, because there's an awful lot of work showing that when when animals or people look at somebody upside down, they don't recognize them very well. So this, I'm a little suspicious of this. On the other hand, this is a very reputable guy. He's done lots of good stuff that nobody has ever doubted him. So, you know, who knows? But the basic idea is correct, that this cell, the cell he's recording from in this experiment, the cell that he's recording from in this experiment is very selective for, in this case, him. Now, obviously, it's not very plausible to imagine that monkeys come born with a cell for recognizing this particular guy. His name is Parrot, actually. It's very unlikely, and so what one must presume is that these cells are formed as the connections to these cells are established as a result of experience with the particular objects they respond to. And a somewhat later experiment from a different laboratory, from a Japanese laboratory that does a small amount of really good work, created a computer-generated patterns, so-called fractal patterns. Here's just some of the ones that they created. And then they make a big population of patterns. And then for a whole bunch of the patterns, they made the animals very familiar with them. What the animals had to do is they had to learn. They would show them a pattern, and then they had to wait 30 minutes, and then they would show two different patterns and say, which one did you see before? And they'd have to respond. So they got very familiar with this set of patterns in order to do this task. And then they have another set of patterns that were generated in the same way that they'd never seen before. If you now record from cells of the inferotemporal cortex, what you find is that for familiar patterns, you will usually find some pattern of the ones that they were experienced with that the cell fires a lot to. This is the firing over time and a number of different tries of presentation. Whereas if you present unfamiliar patterns, they don't respond anywhere near as much. Now, these cells are selective, but not totally selective. For the most part, a given cell usually responds like this to one or two of these familiar stimuli. So they're pretty selective, but not perfectly selective. Now, there are two sorts of ideas floating around for what the neurons representing these meaningful objects is. By the way, there's something I didn't say I should have said. Let me just say it right now, unless I forget again. When you find these cells in any of these kinds of experiments in the inferotemporal cortex, they don't care where in the visual field or where on the retina the objects are presented. The object looks like the right, and they don't care if it's big or small, far away or distant, so on. They just care what its form is, what it looks like. Now, there are two different ways of thinking about what these cells represent. One of them is what I've called the dedicated way, and one is what I've called the distributed way. The dedicated way is very easy to understand. If you imagine that these are just two hypothetical, imaginary figures to give you the idea, George and Harry. George and Harry have different shaped heads. They both have slitty horizontal eyes and thick lips, but they have different kinds of noses. They also have the same kinds of ears. So there's some similarities and some differences between them. Now, if we imagine then that there are cells in the nervous system that code the various features, so various face shapes, various eye shapes, various nose shapes, various lip shapes, various ear shapes, then different individuals are represented by different combinations of these things. One way of thinking about there being cells that seem to respond pretty selectively to George and other cells that seem to respond pretty selectively to Harry is that things are wired up like this. Here's, if we take George here, George has a square head, and so he gets input from the square face shape representation or detector. He has thick lips, so he gets input from the thick lip detector, so the excitatory synapse, and so on. Okay? Whereas Harry over here gets a somewhat different set of inputs, partially overlapping, but partially different. Now, the idea here is that once you have a reasonable number, a reasonable proportion of all the features there are that are all correct for a given individual, the threshold for the cell is such that it will fire. When it does fire, and that wasn't necessarily all the characteristics. You don't necessarily look at every single aspect of the face. You look at you may look at the top of the face, you may see its side views, so you don't see the whole thing. But once this cell here, in the case of the cell for George, once it gets enough input that the cell fires, what it then does is it projects back to all the cells that feed it. So, in fact, the monkey presumably has a kind of a representation in his mind of all the features that lead to Harry, as well as the representation, as well as the firing of this cell that specifically represents Harry. And similarly over here for George. So that's the dedicated neuron idea. The idea that these faces in this experiment or other patterns, other visual patterns in other circumstances are represented by specific cells that are dedicated to them. It wouldn't just typically be probably one cell. There'd probably be a bunch of cells, but not very many. Out of the millions and millions of cells that there would be in probably tens of millions or hundreds of millions of cells in the infratemporal cortex, there would be some modest number that would probably respond to George on the scheme, and another different modest number, but totally different modest number that would respond to Harry. So in this idea, these cells at this level of recognition of individuals, the cells are totally non-overlapping. Some cells respond to one individual, other cells respond to a different individual, or a different stimulus, stimulus configuration, but there's no crossover. Another way that people think about it, what they think might be going on, is that you don't actually have any cells like this at all. Rather, what you have is you have connections between the different cells that represent the features of the individual. So in this case, Harry here has a round shaped face, and he has thick lips, so there's a connection both ways between the face shape, the round face shape, and the thick lips, and so on. George over here has a square head, and so there's a connection between the cell representing his square head and the cell representing his horizontal slitty eyes, and so on. And so, again, because of these mutually excitatory cross connections, it would be the case if you have a preponderance of stimuli, a fair number of the total set of stimuli that go with one of these two faces, that then activates the others because they're all cross-connected, and you get the whole assembly going again. This is sometimes called a cell assembly representation. The difference between these two things don't work so very differently, but the difference is here you've got cells that actually really do represent the individuals in question. Here you don't. Here it's all got to do with the connections between these higher order cells, or lower order cells, however you want to say it, the earlier cells in the system that represent the characteristics of the face, but not the face per se. There is not universal agreement on which of these two ideas is right, but almost everybody, I happen to be personally a doubter, but almost everybody thinks that that's the one that really happens. They think that for several reasons. One reason is they just think it's implausible that you could have this specific set of cells for every different individual or thing you know. I think that's a pretty specious argument because it doesn't really require, you don't really know that many things when it really comes down to it in terms of the numbers of cells there are in the brain. So that isn't a very good criticism. Actually, I guess this is one of another reason that people think this, to think that the scheme over here is the right one, is that when you're recording from the cells, the infratemporal cortex, it's very hard to find. It's almost always the case that if you find a cell, it's very commonly the case, that if you find a cell that responds to one thing, you'll find there's something else too that it will respond to. And that's, of course, much more consistent with this idea than that idea. On the other hand, if you think about it, suppose this idea were right. If this idea were right, there would be just a few cells that would respond to these individuals. There, of course, would be a lot of cells that would respond to the characteristics of the individuals. And there might be some in-between cells, not shown in this picture, that respond to various conjunctions of various attributes of the individual, but are not fully selective for the individual itself. The chances when you're going in and recording at random from the cell, the infratemporal cortex, of actually getting one of these real, totally selective guys, even if they're there, is very small. There might be a dozen such cells out of maybe millions of cells. So it's a very low chance of finding them. If you find a cell that responds to a particular individual, it's much more likely you would be getting one of these cells that are more like this, or that would respond to several different individuals. So the fact that these kinds of cells aren't very often found, or never found very, very rarely, doesn't necessarily mean that they're not the way it's really done. Now, there's a really interesting experiment that was done here at UCLA about 10 years ago. over in the medical school that doesn't really prove the point, but it does bias one in favor of the face cell idea. This was an experiment in which they were preparing patients for surgery, and as part of the preparation process, they had to record, they had to put electrodes in the brain to do various things, and so some scientists were given a half an hour each individual to just record whatever they wanted to record. So they had a whole battery of stimuli. All things that everybody would know, the Golden Gate Bridge, Jennifer Aniston, who is that? Everybody says that. I look at her, it doesn't look like Julia Roberts to me. Oh, anyway. But right, that's what everybody always says. In any case, a whole battery of things that most people are familiar with. And here is one of the cells, and this is a typical kind of picture. This cell responds to Jennifer Aniston looking like that, does not respond to Julia Roberts, responds quite well to Jennifer Aniston again with a totally different kind of picture. Her hair is different. It's obviously a very different sort of a picture. And even responds if you put up a little placard with Jennifer Aniston written on it. Now, this doesn't really prove anything, but it does tend to bias one, I think. It biases me in favor of thinking maybe something like that is right. But as I say, most people don't think so. Most people think it's like that, but the final word is not in. Okay. I guess. Yes.
So, do the proponents of the distributed theory actually believe that there are neurons which code for like specific facial features? Or, like,
well, no, that's a little bit of an oversimplification. Okay. They kind of yes, but it wouldn't necessarily be things that we would give names to.
All right.
But it'd be metric features of the face. God knows what.
Okay, so it would seem like it'd be only.
There probably would be some of things like that, but there would be a lot of others that we wouldn't be able to give a name to.
Okay.
Yeah. That's a good question. Okay. So that's then it for response properties of neurons at various levels of the pathway. And I want to turn now to talking a little bit about modularity. We've talked about the concept already, that there are different parts of the brain that do different things, and we made a distinction between the dorsal stream, the where stream, the ventral stream, the what stream. I want to go into all that a little bit more detail now and actually present you some of the kinds of data that are investigated. So we've already discussed the fact that we have these two different streams, and that the where stream, among other things, responds to movement in particular directions and locations. And so what's the evidence for that? Why do people say that? Well, there are a whole bunch of things. The first, not so the very first one was found, but one of the important kinds of evidence is that if you record from cells in in area MT, which is one of the cells, one of the parts of the brain that's in this where pathway, record from cells, here in MT, the one on the inner surface of the temporal lobe. What you find is that if you pick up a given cell, it will respond to movement in a particular direction. If you have a moving stimulus, it will respond to movement in a particular direction and not others. And the way the stimuli are presented in at least this experiment is there's a whole field of dots, and they're all moving together in some direction. And then the whole visual field is filled with these guys, and they're just all moving in some direction, and you now vary the direction and find out what direction this cell that you happen to be recording from likes. Having found that, if you now move your electrode a little bit in one direction or the other and pick up another cell, you'll find that it also has a selective direction. But it will not be the same direction as this, but it won't be a very different direction from this. It will be a little bit different direction from this, and so on. So you kind of have a map in terms of the angle of direction of movement as you go along this different columns of this region of the brain. So you see that, you say, oh, yeah, it kind of looks like this brain represents direction of motion. It's a reasonable hypothesis based upon this evidence. But how do you know, or the question people who do these experiments ask, is how do you know that these cells firing are really the cause of the monkey or the individual's perception of motion in a given direction? And maybe there's some result of that. Maybe they're not the cause of it. Maybe they're not the actual reason why that's what you perceive. So another kind of experiment that you can do to try to get at that question is to damage the region. And so in this experiment, what was done is, again, you have these dots moving in a given direction, but they're not all moving the same way. in this case here, they're all going the same way. But in this case here, there are more of them going up than there are doing other things. The rest of them are doing something random. But there's still, it's a kind of a net, if you look at it, I'll get to just a second. If you look at it, there's kind of a net drift upwards. Here, they're all going in random directions, so you don't get any sense of motion at all. Yeah?
Is this like a visual system?
Yes. Yeah. Field of dots on screen. Okay, and so you now can ask, well, how much coherence do you have to have to the motion in order for a monkey to be able to indicate what direction he thinks the dots are going in? You train the monkey up, a long time training, and here he is with this manipulanda, which he can move in the direction that he thinks the dots are going. You can then ask, okay, now here's a scale. In a control monkey, there's that amount of, you don't need very much correlation. Only 1% or less than maybe 2% correlation in order to have things looking that way. Note this is a log scale, so 1, 10, 100. So it's quite a low correlation. If you make a lesion of MP, not necessarily removing all of it, but getting a lot of it, then the monkey can still indicate the direction of motion, but it requires much higher coherence in order for it to be able to do it. All right, so since you can damage the region and mess up the monkey's ability to do this, this suggests that this region is kind of responsible for the perception or detection of motion. One last experiment. This one's, hold on. Ooh, all right, well. I don't know if I can get it back again. You saw it move, I hope. I didn't mean to do that. Actually, I can go back a slide. Sorry, let me just see. I think you've got my slide. Come on. No, well, I don't know. It doesn't matter. Let's go just do that. There we go. All right. So the question is, what leads to the perception of motion? Really the firing of itself leads to the perception of motion. Now, if I now I'm going to do it on purpose. If I get this thing to move from left to right, like so, you all get a distinct sense of motion. But there are two, I'll say, did it move? And you'll say yes. But there are two ways you can answer that question. One is because all of you probably did have, unless you're brain damaged, probably did have a sense of motion there. But there's another way you could have done it. You know, before it was over here, afterwards it was over here, so it must have moved. Right? So you can do it with logical faculties. But there's a big difference. In one case, you actually have a sense of it happening. In the other case, you know it happened. You can infer that it happened, but you don't actually feel it. So how do you distinguish these things? Well, it's hard to distinguish, and I'm not sure you fully can. But an interesting kind of experiment that's been done is the following one. So the question is, are these cells the things that the monkey is looking at? So if you look at the pink dots here, if you move the dots up, the monkey sees them, or indicates with this manipulandum that it's moving up. If you move them down, it does what the pink dot says. If you move it to the right, it does what that pink dot says, and so on. So a normal monkey does all this more or less faithfully, correctly. Suppose, however, you now go into the MT, area MT, and find a cell that responds to this direction of motion, the direction down where the red arrow is here. So that's its preferred direction of motion. And now suppose you electrically stimulate in this region so as to depolarize the cells in that region and make them fire. Then you'll have a lot of the cells in this column that likes this direction of motion firing a lot due to this electrical stimulation. And when that does then, you now can do the same experiment over again. You find out that all the monkey's judgments, if you start presenting the stimuli while you're doing this red arrow type stimulation, all of them are biased in the direction of moving what the monkey sees toward that direction of motion. So that suggests that really does suggest that the monkey's brain is looking at, paying attention to, responding to the firing of these cells in making its judgments about motion. So these are the kinds of reasons we say MT is responsible for a sense of motion. Another example of a different kind of an area that specializes for something else is if we go here along the oops, that's not right. If we go along the oh, it is right. Sorry, here's inferocortex. If we go along the what pathway, here's temporal cortex. Suppose we go back a step to region V4. Turns out V4 does a lot of things that suggest that it is specialized for color processing. Not just seeing colors, but doing fancy things with colors. Let me give you an example. Lights out for this. Probably good enough. Okay, this is called a Mondrian pattern. The thing to note about it is there's a green square in the center and a bunch of other colors on the outside. Now, suppose we just show the green square. Fine, there it is. Suppose we now flood the whole thing with red light. Green square turns what color? Yellow, right. We'll talk in a few minutes about why that happens, but it happens. Now, suppose we shine the red light on the Mondrian square. What color is that? We had this discussion before. It's kind of greenish yellow, but the point is it's much more greenish than the previous one was yellow. And what's happening here is one's brain is taking account of the fact that everything has a red cast to it, and is doing an automatic correction and changing its judgment as to what color that is. It's called color constancy. And the thing that's interesting, I mean, that is interesting that the brain does that, but what's also interesting is if you record from cells in area V4, they behave this way. The cells, it looks as though this color constancy computation is being done in area V4, and that's one of the things that V4 is specialized for doing, yes.
Is there a reason that they're like horizontal lines?
Oh, yes. Okay, it's the way these things are made. This is it's a lousy job. It's the best I could do. I think I mentioned to you before I was having computer problems. The way these things are made is there's a whole bunch of very closely spaced red lines, and then the other colors are interlaced as horizontal lines in between and lined up. And the problem is the lineup isn't perfect, so that's why you get some of the funny stuff. I had a program that did it, but it won't work on this projector, and so I sort of did a crude job of it here. We'll see some more pictures like that. The other ones are better. This is the worst one. This is the best I could do. Actually, if I'd wanted to spend a few days programming, I could have done it, but I didn't. I will someday. Okay, so we have these different areas of the visual system that are specialized for doing different things, and these are good examples of this general property of modularity. Now, the existence of modularity raises an interesting question which has not been solved. If you have various characteristics of a stimulus being processed in different portions of the brain, so one's looking at color, one's looking at movement, one's looking at then you have a problem. Some of these areas are not location sensitive, so the what pathway doesn't care where things are. But suppose you have various cues, you've got to put the things that are together in the same place. But if the cells that are analyzing, for example, movement don't, sorry, if the cells that are analyzing what don't know where things are, it complicates things. You've got to get the various analyses, the various kinds of analysis on a given stimulus together on the same stimulus. So here, if you've got two different guys in two different places, how do you, and a lot of the processing is not location specific, how do you do the right thing? How do you keep the properties of this and the properties of this separate, just because they're two different locations? So this is called the binding problem, and there's been lots of ideas about how it works. Nobody really knows. Probably there are a lot of there's a lot of neural circuitry communicating between these different areas, such as the movement area and the color area and the what area and so on, that are doing things that we don't know about and that we haven't worked out. But it's a problem that has not been solved. Okay. Next thing I want to talk about is this thing that I mentioned at the beginning of this block of lectures, and that is that what we see is not what's out there. And we're going to talk about two examples, 2.5 examples. First, we're going to give you some evidence that what we see is based upon reconstruction, some evidence, and then I'll give you sort of another example as well. So kind of 1.5 examples of that. And then we will talk about an example, a situation where it's not just evidence, but where the brain builds one's perception out of whole cloth, which is color perception. So we'll come to that. So first of all, what we see is not what's out there. Okay, now we talked about the retinal ganglion cells, and we talked about the fact that there are on cells and off cells. That was a slight oversimplification, or slight simplification. Really, the cells that we call on cells should be called on-center cells, and the ones we call off cells should be called off-center cells. And what actually happens is this. These cells in the, these ganglion cells in the retina have what's called a center surround organization. Ganglion cells are excited by their favorite input in the center, either light or dark, in the center of their sensitive regions, and they are inhibited almost as much by the same kind of stimulation around their periphery. So if you, for example, have an on cell and you have it just uniformly illuminated, it's getting excited by the light in the center, and it's getting inhibited almost as much as it was excited by the light around the periphery. So the result is that even though this is an on cell, and even though it's being very well illuminated, it fires only a tiny bit, because all the excitation coming from here is countered by inhibition coming from this sensitive surround region. In dark, it's just the other way around. Darkness excites this cell here a little bit, but not very much. But at the periphery, at the border between a light region and a dark region, you have a different game, because there, part of the inhibitory region is not covered up. So, for example, if you have this guy, this is exciting, and this much is inhibiting, but this part here is not inhibiting, because that part there, which is crosshatched, is in the dark region, not in the light region. So now the excitatory central stuff can win out better, and so this cell fires quite vigorously, and correspondingly for a dark, for an off cell on the other side of the boundary. So you get only a tiny bit of firing of a cell that's representing this part of the visual field, only a tiny of an on cell that's representing this part, only a tiny bit here. But here you have the on cells firing very vigorously at this side of the border, and the off cells firing very vigorously at the other side of the border. Now, given that, suppose that we show somebody a picture, we shine onto the retina, or have somebody look at a figure that looks like this, and ask, what is the message that's going to go up the optic tract to the rest of the brain? Well, what's going to happen is you're going to have almost no firing of cells representing this region or this region. The firing is all going to be right around the, almost all are going to be right around the borders. Not 100%, but a lot, 90%, 95%. And you'll have one group of cells that are firing on one side, where it is, so this region is darker than this region. So on this side, on the inside of the border, you have a bunch of dark cells, or off cells, firing. And just outside, you have a bunch of on cells, or bright cells, firing. And so the message that goes up to the brain, the cells are active, only provide information about the outline of this thing. They don't provide hardly any information about the center, and hardly any information about the region surrounding. So, what happens then? But what do you see? Well, you see just that. That's what you see. So subjectively, you get it right, but the message going up to your brain is all wrong, but it's not all wrong, because it provides the relevant information. What the brain is apparently doing is the brain somehow does a calculation that says, in effect, if it is darker at B than at A, which it knows because of the situation on the border, and there are no changes in brightness anywhere else within the object or outside on the outside in the outer region, then it must be dark throughout the whole square. It's an inference. So the brain makes that inference automatically, but then it doesn't just tell you the inference, it reconstructs an image which actually corresponds to what the original illumination was. Nobody has a clue. Well, that's a slight exaggeration. There's very little knowledge of how that's done or where it's represented. It raises the question, are there cells somewhere in the visual system that respond, this response is the way we see things? You have somewhere in the visual system where you have a retinotopic or a visiotopic or topographic representation where you have what is in effect a projection of this image where the cells in the center are doing one thing and the cells on the outside are doing something else in a uniform way, and where you don't just have things going on around the periphery of the object. And the answer to that really isn't known. Not such a hard question. I'm actually disappointed in my visual system colleagues. They don't know the answer yet, but they know. Okay, so I check in with them periodically. I just checked with one of them a week ago. Okay, so the moral of the story is that the brain constructs our mental images of things based on, 1, evidence provided by the sense organs, 2, its own built-in knowledge of how the world is arranged. In other words, if you have something changing here and nothing changing in between and something changing here, then presumably the simulation level is constant in between. That's basically knowledge about the world. That's built into your brain. And then also put in is our memories. Your memories feed into this. And based upon these 3 ingredients together, we build our perception. Deaf grandmother story, don't forget. Okay. That's to remind me to tell you a story which illustrates this point that I'm just making. Quite some years ago now, I was driving my daughter's carpool with these kids in it, I don't know, 2nd or 3rd grade or something like that, at home from school one day. And one of them says, my grandmother lives with us and she really annoys me. She's almost deaf. She's really, very deaf. And you'll say something to her and she pretends like she understands it. And she just answers back, but she gets it all wrong. And why? If she knows she's deaf, why does she just not, just butt out? Don't say anything if she can't hear. She knows she can't hear. Well, she doesn't know she can't hear, because the whole way that everybody's nervous system, including the grandmother's, works is they take whatever evidence they can get and they construct a meaningful thing out of it. And so grandmother heard things. She got the wrong, she didn't hear it right. She didn't construct it right because her evidence was really lousy, because her ears were really shot. But the processes, her brain was just fine. It was just her ears that were at fault. And so she did hear the thing she thought she heard. The only trouble is the evidence coming in was scrambled, and so her brain made the wrong inferences. And so that's a very nice example of these processes going on inside the nervous system. And the same may be true, people have hallucinations. There's probably something a little active that's generating the brain to make a meaningful stimulus, which then people perceive as hallucination. Now that's just a speculation. I know nothing about hallucination. But it would be consistent with the way the brain works. Okay, now our last example here of the brain constructing things is the one that I said I would do, of the brain constructing things out of whole cloth. And that's color. So here is a spectrum. That is blue, that is turquoise, that is green, that is yellow, that is orange, that is red. Most of us, unless you're colorblind, see it pretty much, at least think we see it the same way. We apply the same words to different colors, and we all see different colors as we go across there. But from a physical point of view, there is one continuous variable with no qualitative difference between the different colors, and that's the wavelength of the light, period. And that's the wavelengths that correspond to these various colors. There is absolutely nothing in the physical world that corresponds to the qualitative difference between this and this and this and this and this and this. It's totally a construction of your brain. In a way, I could probably stop the lecture right now because that really makes the point, but we're going to go into it in much more detail because there's some interesting questions to get raised. Now, if you look at the receptor cells of the retina, there are in fact 3 kinds of cells that are wavelength or sensitive. They are the so-called short sensitive, middle sensitive, and long sensitive cells. And they all have very broad sensitivities. So the S cells respond best to stimuli in the short wavelength, but they taper off as they go up here, but they almost all, the middle way in the visible spectrum, they're still responding to about that point there. The middle cells like the middle region best. They don't respond at all to things on the very ends, but they respond somewhat to things almost all the way along, though best to middle wavelengths. And long cells, same story, but off at the right end. But a lot of overlap, very broadly tuned. This is the information that your brain uses it starts with, to produce these perceptions of color, which are so distinct. And in essence, what your brain does is it looks, in order to decide, compute what color it's going to make you think it is based upon the relative amount of firing of the S, M, and L, the short, middle, and long wavelength visual receptors. So blue is perceived if the short guys are responding more than the middle guys are responding more than the long guys. Short, middle, long. Red is perceived if the long are responding much more than either of the other two. Green is perceived if the middles are responding more than the longs and the shorts aren't responding very much at all, and so on. So by looking at the relative amounts of excitation or firing or whatever of the visual receptors, they don't exactly fire in the normal way that we're used to, but the amount of excitation of them, the brain somehow is able to construct the sense of color that you have. Now, given that it does it in that screwy sort of way, there are some funny games we ought to be able to play. Suppose we shine both a green light and a red light on the retina simultaneously. So we've got a green light and a red light. What would be seen? Well, in order to figure out what would be seen, we have to ask how much are each of these three kinds of receptors firing. Well, the middle guys are responding quite a bit to the green. The long guys are responding quite a bit to the green. And the short guys aren't responding very much at all. If we look over here, the long guys are also getting inputs from the reds. So the total amount of firing of the long guys is that amount plus that amount. The total amount of firing of the middle guys is that amount. They don't get hardly any input over here, so it's mainly the green light that's activating the middle guys, and the short guys aren't being excited by either of these two lights, either by the green one or the red one. So now I've converted, using the scale here, I've converted these to numbers as best I could, and then I've added them up. And so the total amount of activity of the short, middle, and long are 0, 9, and 14. That's a ratio of 0 to 6.5 to 10. Put it back onto the scale. And if we do that, then what it says is we ought to be seeing, so we've got short, long a lot, middle, medium, and short not at all. What we ought to be seeing is yellow. Weird prediction. So let's try it. We already know this. We already did this in the context of the Mondrian square. I don't know if the lights out, and that's exactly what happens. So as you ask, these are a bunch of stripes that go all the way across, red stripes that go all the way across. These are a bunch of green stripes that go all the way across that are interleaved with these guys. So in this region, you've got the red and the green interleaved. If you look carefully, you can probably see that. But what happens is, basically, the red and the green fuse in your eye, because your eye fuzzes them up a bit. And so, unless you look at it very carefully and try to focus in on it, what you get is a very clear perception of yellow. And it's because, in part because of what I just told you. But that's not the whole story. It gets more complicated. If we go from the receptor cells, oh, by the way, let me back up a moment. I actually want you to be able to do these sorts of sums. Okay, I can guarantee you there will be study questions and probably exam questions in which in some way you have to be able to do this sort of analysis. It's worth my saying that it's not that I think it's important that everybody go away from this class. That every educated person should know how to do this. That's ridiculous. But what is important, this is one of the few science classes most of you will take, one of the few, or at least one or two biological science classes that you will take. And it is important for you to understand not only the facts and information that this class provides, or a class like this provides, but also to see the kinds of logic and reasoning and mental processes that go on in doing this kind of work. And this is an instance of being able to do that. So it's not that I give a damn that you go away and remember how to do this, but I do want you to have understood it well enough to put it on an exam, at least while you're still here, yeah.
What is white?
What is what? White? We'll come to white in a minute. Okay. Now the picture at the level of the ganglion cells, which are the next step, receptor cells, bipolar cells, ganglion cells. Well, the ganglion cells is different. And the picture for the ganglion cells holds quite a way into the brain. So once we're talking about the ganglion cells, we're also talking about the brain. There is circuitry between the receptor cells and the ganglion cells that changes the description of what's going on around. If you record from ganglion cells and shine colored lights on them, they again they are what are called red. They're what are called opponent cells. So there are basically 4 kinds of color-sensitive retinal ganglion cells. One are the red cells, which are inhibited by green. So they're called red-green opponent cells. So the red cells are excited by red and inhibited by green. There are green cells, which are excited by green and inhibited by red. There are yellow cells, which are excited by yellow and inhibited by blue. And there are blue cells, which are excited by blue and inhibited by yellow. And so if you apply a yellow light and a blue light of the same intensity, all sense of color for these guys will cancel out. And that's how you get white. I'll show you that in a minute. If you do the same thing up here, it's not equal amounts. You have to have slightly, I forget which one has to be brighter. One has to be a little brighter than the other, but they again cancel out. If they're both equally bright, you get a vague sense of yellow. Because of the way the circuitry works, it activates these guys a little bit more than these, and so you get this slopping over. So it's a different picture. Now, if you put that together, you get one version of the color wheel or color square or whatever you want to call it. Now, let me say something to be careful. Most of you are probably much more familiar with combining pigments than combining lights. Here we're talking about combining lights. So, for example, you probably all know that yellow, what color do yellow and blue make? Green, right. That is true for pigments. It's because with pigments you're subtracting, you're not adding. Pigments soak things up, so it's a whole different thing. I believe I have a question on your study questions that will allow you to sort of understand how that works. But right now we're talking about light, not pigments. Okay, so forget what you knew. Don't, if you answer questions based upon what you know from what we all know from mixing pigments, you'll get the wrong answers. In any case, so we have this sort of color combination chart. We have red, yellow, green, blue. Those are the pure things that the various four types of ganglion cells respond to. If we present yellow and blue together, they inhibit one another and we lose a sense of color. If you present red and green together in the proper proportions, the same thing happens. But going the other way is not true. If you combine red and yellow, you can have, that's fine. They're independent systems. So you have red cells firing and yellow cells firing. And so, well, 2 things happen. First of all, you get a sense of orange. But everybody, if you look at orange and say, describe orange, well, you'll say it's reddish yellow. Okay, so orange is reddish yellow. Whether orange is a distinct quality, distinct from reddish yellow, is something we'll discuss a little bit more next time. Time to stop. Finish with this next time. Sorry. I have a quick. I don't think they include brown, but they do include some colors that some of the intermediate, some of the other kinds of colors. So that's a little bit of a. And I don't know if you get brown by mixing. You can get that mixing. I don't know if you get that mixing white. I'm not sure about that. Yeah, I'm not.
Especially because you said there's no white. It's just colors.
Just colors.
How do you get? There's no actual color white. It's just color red.
That's right. Yes, by not very much red. So if you go, I think if you take red and green, and if you bring balance, so you've got colorless, and then you move it a little bit in the direction of red, I think you might get pink. Yeah.
Then brown is something else.
I think brown. I can't answer the question for brown.
Okay, so I tried to download the study questions last night, but what downloaded was just another version, which is the second copy of each slide.
Say that again.
I tried to download the study question, the neuroanatomy.
So I from the website or from what I sent you on. So I believe that I sent you everybody directly by email both
okay.
times.
Right.
But I did post them, so maybe I put the wrong thing there.
Yeah.
Okay, I'll check.
Yeah.
I'll check. Thank you very much.
I just have a quick question. You mentioned that by the brain, but don't you have to have the stimulus to stimulate?
You have to have the different wavelengths, but there is nothing in the physical stimulus that will explain the boundaries between one qualitatively different color and the other and the next qualitatively different color. The difference between green and yellow is simply a quantitative difference in the wavelength. And if you look at physically, there's nothing special happening. If you don't look inside the brain, there's no way of knowing that there's something different, that you're going to perceive something different for whether I don't have it up there anymore, but no. Right.
Actually, I already picked up mine.
So it's here.
Right.
Just no. Come on. No, maybe I'm not going to do it. I think I'm just. Never mind, I can't do it now.
So
it's crashing on. Oh, I see. Wait a minute.
So the only.
Oh shit. Excuse me. Please, baby. I might have just woke up. No, I'm okay. I can't do it for you now, though. But yeah. So, Angie, let's get out of here so the next person can.